Hello, we're going to take a look at a specific type of enzyme called restriction enzymes. Okay, these are enzymes that are found naturally in bacteria, and they have the property of cutting DNA at a specific nucleotide sequence. They're actually occur naturally in bacteria as defense against viruses that infect bacteria, but molecular biologists can use them to cut DNA at specific sequences and use those for biotechnology and recombinant DNA. So let's see how restriction enzymes work. Okay, so here we have a sequence of DNA. It can be any DNA from any source, doesn't matter. Remember, DNA only the sequence matters. So here we have a restriction enzyme called EcoR1. So EcoR1, let me move it down a little bit. EcoR1 stands for E. coli restriction enzyme 1. And it comes from the bacteria E. coli. And it's looking for this sequence. So when it recognizes a sequence in DNA, GAA-TTC, 5' to 3', it will cut there. Now, there are different ways that it can cut, that restriction enzymes can cut. This one happens to be, produce sticky ends, and we'll see what that means in just a moment. So it will scan along this piece of DNA until it comes to that sequence GAA-TTC. Well, here it is. So it's going to cut at that sequence, and it cuts in this pattern here. Okay, so it cuts between the G and A here and the G, A and G on the opposite strand. So it cuts. Here it is cutting the DNA. And now it cuts the phosphodiester bond and the two strands separate. It leaves what's called a sticky end. It's called sticky because you see the single-stranded overhang here has hydrogen bonds. Now, there are other enzymes called ligases, we've seen that with DNA replication, that can seal this nick back up. Okay, so any DNA cut with with a particular restriction enzyme can be religated. So it's religated here, but we don't have the ligase, DNA ligase, so now the DNA is cut. Let's look at another example of a restriction enzyme. This one is called SMA1 from Serratia marcescens, and it recognizes the site CCCGGG. So it's going to scan along this DNA until it finds that site there, and it will cut like this. And this is called a blunt cutter. It's blunt because it doesn't have any 5' or 3' overhangs. Now let's go back to our original piece of DNA. We'll keep this one here. And EcoR1 is still scanning along. Are there any other sites? Scanning along here. And another one, GAA-TTC. So it's going to cut there as well. Again, it cuts in that same pattern. Okay, so no more sites in there for EcoR1 in this DNA. So let's see how many fragments this has generated. Okay, so 1 fragment, 2 fragments, 3 fragments. Let's go back to the small one. Okay, this DNA here, and see if there are any more sites that it recognizes. CCCGGG. Okay, each different restriction enzyme recognizes a different sequence. So are there any more along here? No. Okay, so this generates 2 fragments of DNA. So we have 2 fragments generated, small 1 from this DNA, and 3 fragments from EcoR1. So let me pull that one down here. Okay, and again, it doesn't matter the source of DNA. It can be from humans, it can be from yeast, it can be from any source. So restriction enzymes, each one recognizes a specific DNA sequence. And there are hundreds of different restriction enzymes that are known. So we looked at EcoR1. Let's pull that one down here. Okay, that recognizes GAA-TTC. And notice the complementary strand, GAA-TTC, going in the opposite direction. And small 1 recognizes CCC-GGG.